order. Order is now resumed.
Thank you. Good morning, Mr. Commissioner. Today we'll be dealing with technical report number 6, the cumulative impact assessment with David Marmrick. But before we get started, there's a couple of housekeeping matters that are outstanding. In the July marine hearings, Dr. Tim Parson was a witness, and a number of documents were referred to by him, and Ms. Gertner asked him to produce those articles to us over the course of the break, which was done, and that was circulated to all parties on just after September 1 when it was sent to us. So I'd like those 3 articles now marked as exhibits. They are, the first one would be something that I can't even pronounce, so scientific values as indicators of trophic position and competitive overlap for Pacific salmon, which is an article by Welsh and Parsons in 1993. That would be the first exhibit.
We marked as exhibit 1892.
Thank you. And the next article is sea surface temperature and the preseason prediction of return timing in Fraser River sockeye salmon, an article by Blackburn. That's it.
1893.
And the last one is locations of marine animals revealed by carbon isotopes, an article by Mackenzie Palmer et al. Sorry, I don't see the year.
1894.
Thank you. And then coming out of the hearings in August, the marine hearings in August, a request was made of Sergio DeFranco by Mr. Rosenblum, which was followed up in writing and circulated to all parties as well. That is contained in a response from Canada is in an email to me and Ms. Tsurumi, and it has some text provided by Mr. DeFranco as well as a table of figures which is attached to the email, and that would be, since it's pulled up, could be marked as the next exhibit.
1895.
We're still waiting to get it on the screen. So we have today...
just before you get underway, Ms. Baker, I just want to welcome to the hearing room a group of students, I think perhaps 30 in number, who are here from Simon Fraser University, and they are students in the Resource and Environmental Management faculty, and so we welcome them here today. Thank you very much.
Thank you. Our witness today is David Marmrick with ESSA Technologies, and I'd like to have him sworn, please.
You solemnly affirm that the evidence to be given by you to this hearing shall be the truth, the whole truth, and nothing but the truth?
Yes.
Would you state your name, please?
David Marmrick.
Thank you.
Are the lead author of Technical Report 6, Fraser River sockeye salmon, data synthesis and cumulative impacts?
Yes.
Thank you. Could I have that marked as the next exhibit?
1896.
Thank you. And there was an addendum done to this report, which has actually already been marked as an exhibit in these hearings. It's been marked as exhibit 1575, if that could just be brought up. Thank you. That's the addendum that was prepared?
Yes.
Thank you. And lastly, you prepared an errata sheet on September 13 to correct a few typographical errors and clarify a number of things in the report. It's a 1-page document. It's on the screen now. Do you see that?
Yes.
Thank you. And that's the errata sheet you prepared?
Yes.
Thank you. I'll have that marked, please.
1897.
Mr. Commissioner, I would like to go over a little bit of the background of the witness, but I have also previously circulated the areas of expertise I'd like to have the witness qualified in. I circulated this to all parties in advance, and I think I've given a copy of this to you as well, so you can follow along. So the areas of expertise I'd like to have him qualified in are aquatic ecology, including the effects of human activities on aquatic ecosystems, fish habitats and fish populations, environmental impact and ecological risk assessment, adaptive management, experimental design, decision analysis and modeling, and technical facilitation of interdisciplinary scientific workshops. And now just to review some of your background, you are an aquatic ecologist and you're the president of ESSA Technologies.
Yes.
And you're also an adjunct professor at the School of Resource and Environmental Management at Simon Fraser University.
Yes.
sorry. Mr. Marmrick's CV has already been marked in these proceedings, and I'll just have it pulled up. It's Exhibit 566, that's just to follow along. Mr. Marmrick's CV was marked in the course of the technical report number 3 hearings. Now, you have spent a number of decades working in areas like simulation modeling, ecological risk assessment, is that right?
Yes.
Aquatic ecology,
Yes.
experimental design, statistical analysis,
Yep.
integration of large-scale research and monitoring programs,
Yes.
adaptive management and decision analysis.
Yes.
Right. And you've also applied some of those skills working with humans in facilitation and team leadership, is that right?
That's right.
Okay. I'd like to ask you a little bit about some of your past experience. I'm not going to spend too much time on it. It's written in the CV, but just in terms of the work that you've done, can you talk to us a little bit about the work you've done in the Columbia River and the path process there?
Sure. Starting about 1993, I was asked to facilitate comparisons amongst different simulation models that were being used to forecast the survival of endangered Chinook salmon, both in the river and over their entire life cycle. And that work sort of morphed into something called PATH, the Plan for Analyzing and Testing Hypotheses, which involved about 12 agencies over about 6 years, looking at the question of whether it was better to barge salmon down the Columbia River past the Snake Dams and Columbia River Dams, 8 of them, or whether it was better to breach the 4 Snake River Dams. So that was a very controversial topic involving fairly adversarial circumstances, and our team from ESSA basically led both the technical facilitation of that as well as integration of models and publication of various results, which were extensively peer-reviewed by that interagency group as well as reviewers before they're published in journals.
Right. And have you done any work in the Fraser River Basin?
Yes, going back to the times of the Green Plan in the early 1990s, we looked at fate and effects of pulp mill effluents, did various work on the Fraser as part of the State of Environment report, that BC's first State of Environment report in 1993. Going back even earlier in the 1980s, we looked at various harvest management questions, return of Fraser River stocks for in-season management, and more recently, of course, work with the Pacific Salmon Commission on the Fraser sockeye decline last year in June 2010.
Thank you. And you've, of course, authored numerous peer-reviewed publications, is that right?
Yes, that's right. They're listed in the CV.
Okay. That's right, pages of them, and a number of, over 100, I think, technical reports as well.
Yes.
Mr. Commissioner, I asked my friends to advise me if they had any difficulties with the expertise that I proposed to have him qualified in, and I have heard nothing from them, so I propose that he be qualified in those areas.
Yes, thank you, Ms. Baker.
Thank you. And the report that's now marked as Exhibit 1896, the Technical Report 6, had a number of different authors involved. Some of them have already had their CVs marked in these proceedings, and those would be, just for the record, it would be Mark Nellix.
Katie.
Anyway, sorry. Catherine Bryan, Brian's CV is Exhibit 564. And Catherine Wykowski, her CV is Exhibit 570. The CVs of the authors in these proceedings, so I'll do that now. The first one would be Darcy Picard. That could be pulled up. Thank you. Now this is the CV of the Darcy Picard, who worked on this report with you.
Yes.
Have that marked, please.
1898.
Thank you. Next one would be Liz Martell. And this is similarly the
Yes.
CV. Thank you. Have that marked, please.
1899.
The next one would be Clint Alexander. again?
Yes.
Thank you. Have that marked, please.
1900.
Thank you. Lauren Grieg?
Yeah, it's Greg. Yes.
Greg, sorry.
It's fine.
Thank you. This is his CV.
Yes.
Have that marked, please.
1901.
And the last one is Carl Schwartz.
Yes.
Thank you.
1902.
Thank you. All right. I'd just like to have you give us an overview of the steps or the components, I guess, that you took in creating the report that's now marked as Exhibit 1896. If you can just describe that in a summary way, and we'll get into it in a bit more detail, but the components or steps that you took in creating the report and coming to the conclusions you came to.
So basically 7 steps. So we first developed the approach that we were going to use for the qualitative and quantitative analysis of the evidence. Then we had a workshop November 30th and December 1st of last year with about 30 people, both authors and reviewers of the Cohen Commission reports. The third step was getting as much of the data as we could from those authors on the various stressors, potential stressors affecting sockeye, as well as the productivity data, and organizing that data into a relational database for further analysis. The fourth step was doing a retrospective ecological risk assessment or cumulative impact assessment based on the Cohen Commission technical reports that were relevant to that, and additional evidence from the PSC report on sockeye decline. And then the fifth step was the quantitative statistical analysis, which was in support of that synthesis of evidence, looking at alternative hypotheses about what sets of stressors might have affected which life history stages and ultimately overall life cycle productivity. And then we wrote the technical report and revised it in response to the fairly extensive reviews. And then the last step was the addendum on aquaculture that you just mentioned earlier.
Okay. And can you identify for us what your key conclusions were as a result of this work?
Sure. So the first is that before attributing causality, you need to look at the overall pattern of change in sockeye productivity within both Fraser and non-Fraser stocks. And section 4.1 in our report summarizes the work from Peterman and Dorner and others, Skip McKinnell and so on, about what that pattern is. So that's, I think, the first conclusion. The second one is, in terms of the primary factors responsible for the long-term declines in overall Fraser sockeye productivity and the 2009 low returns. So we concluded, first of all, that marine conditions interacting with climate change during the coastal migration stage were the likely primary factors for the long-term decline over the last 20 years in Fraser river sockeye productivity, and that marine conditions were likely to be the primary factor responsible for the poor returns in 2009 in both Strait of Georgia and Queen Charlotte Sound. With respect to the returning run of spawners from the mouth of the Fraser back to the spawning ground, climate change and en route mortality has definitely affected harvest and escapement, but not productivity measured as recruits per spawner, because that recruitment already includes harvest and en route mortality. It's basically escapement plus harvest plus en route mortality. So that did not affect the overall trends in sockeye productivity. Other possible primary factors in the productivity declines include predation on adult sockeye as they come back to the mouth of the Fraser, and climate change in the early life history stage from egg to smolt. We were not able to draw any conclusion on diseases because of lack of data on the exposure of Fraser sockeye to diseases. And disease transmission from aquaculture, we concluded, was either unlikely or a possible primary factor, depending on which of the two aquaculture reports one uses as evidence. And all the other factors we consider to be unlikely to be primary factors responsible for the overall decline in productivity, for example, many of the freshwater habitat factors, though they may well have contributed to changes in some stocks in some years. So, for example, Delayed density dependence appears to have been responsible for some declines in productivity in the Quesnel sockeye stock in some years, but was not responsible. It was not a primary factor responsible for the overall decline across all the stocks. And finally, there are many gaps in the existing information which make this whole process difficult. So both assessing the exposure as well as the correlation of those exposures with changes in productivity, as well as having life stage specific survival and condition information. So that led to some of the recommendations that we have.
Okay, thank you. Now, you've identified that when you prepared this report, you looked at the technical reports prepared for the Cone Commission, and you also looked at the PSC report in June of 2010?
Yes, we did.
Okay. Did you do any independent research for this report?
Well, some of the things that I just mentioned were independent in the sense that they hadn't been done before, and we weren't picking them out of the existing Cone Commission reports or the PSC reports. So developing our approach to retrospective ecological risk assessment, that was novel, although based on existing published methods. The quantitative statistical analyses were new, and the synthesis, both within and across life history stages and going across all of these reports, was new research. And our recommendations really built on what was already in those reports, but we added some of our own ideas.
And did you independently assess the validity of any of the technical reports prepared for the Cone Commission?
No, we didn't, because we weren't asked to do that. We did carefully examine the methods that each of those authors used, and we looked at the reviews. It was the responsibility of the reviewers to review those reports.
The reviewers being the reports that you see attached at the end of each technical report?
Right.
Okay. And then in the technical reports, if knowledge gaps were identified, would those knowledge gaps then carry forward into your reports? For example, were you asked to address any knowledge gaps that had been identified in the technical reports for the Cone Commission?
We were not asked to fill them, but in the sections where we discussed what we need to know better, and in our final, I think section 5.2 on recommendations, we carried forward some of the recommendations from those reports.
And just a kind of background piece, I'd like to get into the report, and have a figure in front of us as I ask these questions. Sorry, it's the actual page number, not the PDF number. So, yeah. There. So there's a figure there on the screen which you call the cumulative stress model, and I just want to have that up there and then ask you the question. Your report is called Data Synthesis and Cumulative Impacts, and in looking at the cumulative impacts on Fraser River sockeye, were you able to assess first how the stressors within each life cycle combined in a cumulative or an interactive way to create specific impacts on the fish?
Actually, if you wouldn't mind, I'd prefer to have page 18 up here to talk about that question.
Sure.
We can come back to this page later, but I think this is a better figure for the question you're asking within each life history stage.
Right.
So
this is figure, just for the record, 3.3-1.
Right.
It's called the conceptual model of the life history of Fraser River sockeye.
So this is complicated, though one reviewer thought it could be more complicated. Anyway, what we did is we went through all of the technical reports and looked at the candidate stressors affecting each life history stage. And some of those reports were focused on particular stressors, like contaminants or aquaculture or disease or predators, whereas some other reports were focused on particular life history stages. So there was a report on fresh water, which looks at egg, alevins, fried par, and smolt, things you see at the top of figure 3.3-1. And there was a report on marine conditions, which looks at the post-smolt part of that figure. So we basically built up this conceptual model of the candidate stressors affecting each life history stage from those reports and from the workshop. And some of the reports, particularly those that looked at life history stages like the freshwater report and the marine report, also looked at some of the potential interactions amongst these factors and how they could combine. And also the en route mortality report looked at combined interactions like temperature and pathogens, disease, harvest. all combining. So the main thing was we tried to list all the plausible mechanisms and then consider how those might have interacted, although we don't actually have very little hard evidence on how they interacted.
One of the things that we were looking at, I guess, or what the title suggests, is that you're going to be able to actually assess how a stressor in one life stage could impact those fish as they move through their life cycle. Are you able to do that, or were you able to do that?
So only in a conceptual way or a theoretical way, in the sense that the data don't really exist to sort of carry forward from each life history stage the changes in survival and condition. What we did do, though, is in the analyses we looked at, particularly the correlational part, we're looking at factors which occur within each life history stage and considering how they ended up affecting the overall life cycle productivity. So to the extent that there is some correlation there, let's say that as productivity declined, some particular stressor went up or increased, you're looking at how an effect within a life history stage basically propagated to affect the overall life history or the overall life cycle productivity.
But what we're not able to do, I take it, is see how these sort of the 1,000 cuts, the small assaults on the fish as they go through their life cycle, maybe have a greater impact than the whole. So you could see what a primary impact might be, but you may not be able to understand how a number of small non-lethal effects would have a cumulative effect over the life history.
You don't know that. You don't know that very well, except for those life history stages where you do have some estimates of survival within it. So, for example, if you take the freshwater life history stage, we have 9 of 19 Fraser River stocks where we do have some estimate of survival from spawners to mostly fry, in a couple of cases smolts. And we can look at the patterns over time for that life history stage, and generally speaking, they haven't gone down. So we can say for that life history stage that the cumulative effect of all the factors operating on at least those 9 stocks, at least to the fry stage for 7 of them, doesn't appear to have negatively affected their survival or caused a decreasing trend in survival over the last 20 years, which is the period of interest. So that's the power of having data that discretely summarizes the survival within each life history stage, because you are effectively looking at what's the cumulative effect of all those things, at least up till that point. Now, there could be a delayed effect, so an animal may acquire some disease in that stage and survives fine, there's no trend there, but later on, when it gets out to sea, that could end up affecting its survival. So again, you can't really distinguish that unless you have better estimates of survival at each life history stage.
And is that something that you are able to do on the evidence available today, now?
No, only, well, to a limited extent, in the sense that, as I just indicated, we have data, for example, for the 7 stocks where we measure fall fry, we know survival from spawners to fall fry, but then after that, we're basically going from fall fry all the way back to recruitment. Now, that's a pretty big box. That includes downstream migration, coastal migration, returns. So we haven't been able to distinguish that, with the exception of a few acoustic tag studies, but are only for a few stocks here and there. So that's the gap.
Okay. And that impacts our ability to understand these cumulative impacts as we're going through the life cycle.
Yes, I think the key way to think about this is that the first thing you want to try to determine, this is what a lot of work in the Columbia River has done with pit-tagged fish, for example, is in which life history stage is the bottleneck occurring? And then what are the factors most correlated with that decrease in survival? And you need some contrast, either over space or time, in those stressors to be able to deduce which of those factors are most likely.
And we're not able to do that on Fraser River sockeye, is that what you're saying?
Well, we've done the best job we can with the data we have.
No, I'm just trying to identify that there are a bunch of data gaps that prevent
Yes.
you from doing that full analysis.
That's right. You can't do it as well in the Fraser as you could in the Columbia because of the data that you don't have.
Right. Okay. Going back to figure 2.3-1, which is on page 10, the first one I asked you to look at.
Yeah.
What does the dotted yellow line indicate in the A1 and the A2?
So this is a conceptual figure. On the y-axis you have a measure of cumulative stress, and if you get to 1, you're dead. And on the x-axis you have the different life history stages. So and these are just different pathways that an individual salmon might follow. So A1 is a fish that experienced lots of stress as fry. Perhaps there wasn't enough food, or perhaps there were bad environmental conditions. So it almost died, but not quite, and then got all the way through to the adult stage, and then experienced some other stress, maybe a predator, maybe some disease, and died. A2 represents a situation where this was a fish that survived that early life history crunch, whereas most of its brethren died, and so there was less intraspecific or within species competition. So as they went down to the mouth of the Fraser, they actually found there's lots of food to eat because there were fewer competitors around. So there's actually a decrease in stress. So you can have these kind of compensatory effects that occur between life history stages. They're not always necessarily additive or cumulative or synergistic as you go through life history stages.
And you talk about that in your report. At some point you talk about compensatory reduction, and this is an illustration of what you were referring to?
Yes.
Okay. Page 22 of your report, you talk about a weight of evidence approach that you use to assess whether a stressor or a factor made a substantial contribution to the decline. Can you explain that weight of evidence approach?
Sure. So this is what we were talking about when we said we adapted a retrospective ecological risk assessment framework. So we went through basically 4 sets of questions here, and I'll just read them because it's simpler. The plausible mechanism: does the proposed causal relationship make sense logically and scientifically? Is it possible that contaminants could harm fish? Yes. Is there evidence that sockeye populations are or have been exposed to the causal factor? So another example there would be, we have some data on contaminants regarding their exposure and how that's changed over time. Correlation consistency. So is there evidence for association between adverse effects in sockeye populations and presence of the causal factor, either in time or space? So in the case of contaminants, to continue that example, in general, contaminants did not increase as sockeye productivity decreased, which suggests there is not evidence for an association. So that's the kind of question we were trying to answer. And then the 4th category is other evidence, which can be supportive. So things which say, well, are there certain thresholds which suggest that the exposure level went above those thresholds? In the case of contaminants, that report specifically looked at thresholds, hazard thresholds they called them, for each of the measured water quality and sediment parameters. And then specificity, this is where if there's a particular kind of effect in the population that's caused by exposure to a certain stressor, so you might say there's a certain kind of physiological response to a certain contaminant, and you can then look for that response if you have those data or if there have been experiments on them. And experiments in a laboratory or in a field are also quite helpful in confirming the causes of things. So the experiments that were done in the field, for example, on en route mortality by Tony Farrell and others, where they put fish through a kind of exercise machine and see at what temperatures they die, confirms that certain temperatures kill them. And then removal of the stressor led to an amelioration of the effects in the population. Well, that relies really on some contrast happening in that stressor. So those are the categories of evidence that we looked at, and then organized them in the decision tree, figure 3.3-3.
That's on page 24. All right, can you, as we wait for this to get settled on the screen, can you explain how you applied that weight of evidence approach to this tree?
Sure. So basically takes those questions that I just outlined and goes through asking whether a given factor or hypothesized stressor passes various tests. So the first case is, is the mechanism plausible? And in almost all of the cases in all the reports, the answer to that was yes. The only exception was in the Noakes report, he felt it was not plausible that waste from salmon farms could have an effect. Then we move to the exposure question, which I just described for contaminants. And here we're addressing, are there data by which you can assess changes in exposure over time or over space? And for many of the hypothesized stressors, we didn't have exposure data. And or I should say, for one of them we had no data, and that was for pathogens, and so no conclusion was possible. So the middle box there, when it comes to exposure, we had exposure data, but it wasn't likely that the fish actually got exposed to those stressors. That would be the case for something like mining or small hydro, where there are so few mines or small hydro facilities within the Fraser Basin that it's very unlikely that sockeye spawning and rearing habitats were exposed. So if you get past that set of questions, you then follow the yes box and you come down to, okay, so it looks like there was some exposure. Is there any correlation or consistency? And I ran through an example earlier for contaminants where in general the answer to that was no. There was not consistency in the change in the stressor and the change in productivity. Now, in some cases, we got through that box and down to, yes, it looks like there was some correlation that was consistent with the hypothesis. And so we moved down to the bottom box and other evidence. And so that's where the climate changes and changes in marine condition ended up being either possible or likely factors for some of the life history stages. They got all the way down to the bottom box. And the predators, as far as returning adult salmon, there was some exposure data that looked like some predators had increased over time. And so it looked like it might be possible, but there really weren't good correlation analyses. So we ended up at the bottom without enough evidence to say anything other than it was a possible factor.
When you speak about contaminants in this example, are you referring to the contaminants that were measured in technical report 2, or are you also including the contaminants that Don McDonald identified as being unmeasured, for example, endocrine disruptors and emerging contaminants?
Just the ones that were measured. For things that aren't measured, we would end up in the same place as disease of no conclusion possible.
And again, your focus throughout was to look at primary driving factors, is that right?
That's right. What are the primary factors responsible for driving the overall long-term declines in productivity of Fraser River sockeye, productivity over the last 20 years? And we also looked at non-Fraser sockeye to help distinguish amongst those hypotheses. So some of those that are unlikely, fell in the unlikely box to be primary factors, they could still be contributory factors.
And the non-Fraser stocks, was that the work in technical report 10?
Yes.
All right, now what does this kind of work tell us, or this kind of analysis tell us about each life stage then?
So again, we're looking at the relative likelihood of each factor within each life history stage being a primary driver of the overall declines in Fraser River sockeye productivity over the last 20 years.
And this kind of analysis, does it allow us to tell us, does it allow you to tell us anything about the entire life cycle of the fish?
So because our primary response variable is the overall productivity of Fraser River sockeye across these 19 stocks, we are looking at correlations between the stressors and the overall life cycle productivity. So as I said earlier, it's still, our conclusions within each life history stage do in fact still relate to the overall patterns over the whole life cycle. And as I said earlier also, we do have data for juvenile survival per spawner for 9 of those 19 stocks. So that also provides something about what happens within the life history cycle.
Just to go back in the report, page 14, you talk about, there's a section in your report 3.2 that's titled Unknowns, Unknowables, Knowledge Gaps and Data Limitations. And I know you've talked a little bit about some of these already, but Just at the bottom of the page, you talk about the evaluation of alternative hypotheses and a couple of principles that underlie that analysis. Can you explain those principles?
Well, the first one, sort of general principle of science, is that you can only reject hypotheses or provide evidence against hypotheses. You can't confirm that they're absolutely true. And those for which you have less evidence against them are the ones that become more likely. And the other part there is that correlation does not represent causation. And so we have to be very careful which variables we use to look at correlational patterns. There has to be a reasonable, plausible mechanism before you include a given variable in a correlation analysis. Now, if you find out that, as the example I had earlier on contaminants, that there is not a correlation, that suggests evidence against that hypothesis.
And in terms of data limitations in the evaluation process, were those identified by you?
The data limitations, at the end of each section, we have things we need to know better, and then we summarize those in section 5.2.
Right. And what are the main, like the dominant limitations that you can, or the ones that sort of stood out for you as being the most problematic?
So the first I already mentioned is that when you want to determine at which life history stage bottlenecks are occurring, it's really helpful to have information on survival through each of those life history stages, and also the condition of fish through each of those life history stages. Now, you can't get that perfectly, it would be too expensive, but we could certainly have more information than we currently have. And then the second is, as we've just gone through that decision tree figure, there's gaps in the information on exposures and a shortage of quantitative analyses of correlation and consistency, which make it hard to get all the way down through that tree. You know, an example would be if we had information on diseases, we could say a lot more about the likelihood of that stressor actually being responsible for some of the declines that we've observed.
I think that covers off most of the knowledge gap points I wanted to talk to you about. But what about the unknowables? Page 16, you have you describe a challenge as the third challenge is unknowables. What are you trying to describe there? What's an unknowable?
First of all, it's hard to know exactly how a salmon dies unless it ends up in a fishing net, a predator's stomach, or there's some sort of massive fish kill like happened in the Cheakamus River a few years ago with caustic soda spill. So you can really only infer how a fish died indirectly by looking at strong contrasts across time, across stocks, and across space. And so ideally you have contrasts in survival, and we have a lot of that because productivity trends have varied over time and over space. And so we then can look at the contrasts in stressors. But you're never going to know ultimately exactly how those fish died, even if you could measure all the stressors, and you can't. You're never going to have, you know, for all the coastal migration period, full knowledge of all the predators, competitors, food supply, contaminants, temperature, conditions exactly. So that's essentially unknowable. There is incomplete information. And so you're going to have to make inferences based on contrasts. And you also it's really unknowable exactly how all of those stressors ultimately combine to hit that 1.0 mortality part on the graph we were looking at earlier.
The bulk of your report is where you examine each of the different life stages, and you've broken them into 5 different life stages. Do you have the same degree of confidence in your conclusions for each life stage, and if you don't, how would you rank your confidence in the different life stages?
On a relative basis, with the highest level of confidence first, and going down to the least, and it'd be interesting, people might debate this, but I would put from the mouth of the Fraser to the spawning ground at the highest level of confidence. They've got quite accurate estimates of survival and of a lot of other exposure factors, temperature and disease.
That's life stage 5?
Life stage 5. And then next I would put egg to fry, because we do have, as I mentioned, 7 stocks where we have egg to fry survival and 2 where we have egg to smolt survival. We actually have data there. The next I would put that would be life stage 1. The next would be life stage 3, the coastal migration, because there are data on catch per unit effort, for example, in the Strait of Georgia, which is sort of an index of abundance and kind of like an index of survival. Then I would put the smolt out-migration stage, life stage 2, that is from the time smolts leave rearing lake to the time they get to the estuary. We really have very little data on that. There are a few stocks with acoustic tags, but not many. And then the last one with the least level of confidence would be stage 4, which is the growth in the North Pacific, where they're out there for a couple of years, and we really don't know very much about what they're exposed to and what happened to them until they start coming home.
Okay, thank you. I want to go through your life stages, starting at 1, going to 5, well, not in the order of confidence. So the first one that you referred to, of course, is the egg to fry, which begins at page 39 of your report. And you prepared in your errata sheet some comments on what the other evidence column means here. And I'm wondering, so the conclusions are actually on page 48. I'll just identifying where the chapter or section begins. So if we go to page 48, you've got the conclusion section, which is a table summarizing some of the points that we'll be discussing. And in your errata, you've identified table 4.2-1 as having some changes. So I'm just wondering if you can relate those errata comments to the table. It's a bit difficult with only one screen.
No, that's fine. It's probably fine to just leave the errata up. Or I can read from it. You can put the other table up. But basically, as we just talked about on page 22, the other evidence column refers to the 4th set of questions in that decision tree. And so when we say no in that table, if you go back to page 48.
Thank you.
Yeah. Oh, that's good. So when we say no, that indicates that the other evidence was not available for the listed stressor. In other words, thresholds, experiments, and that sort of thing. It's hard to do an experiment on the proportion of a watershed that's been forested. You can't fit it in easily into a lab. So there were not information available for those stressors where it says no. So yes means that other evidence was available. For example, on contaminants, water use. There are some lab studies that have been done. There have also been lab studies done mostly on hatchery fish for diseases, except we don't have the exposure data. So yes means that other evidence was available for listed stressor from those reports and provide additional support for the hypothesized stressor. Against means that other evidence was available and was contrary to the hypothesized stressors. So for example, there were detailed studies done on the Chaco large hydro, which indicated that it was not likely to have had significant effect on the overall declines in sockeye over this period of time. And then mixed means that other evidence were available, some supporting and some negating the hypothesized stressor.
Okay, thank you. And the likelihood column, can you just explain for us what you're assessing there?
So when we went through that decision tree, this is the conclusion that we came to regarding the, and it's in the first sentence of the caption of table 4.2.1. It's the relative likelihood that potential stressors encountered by Fraser River sockeye salmon during life history stage 1, including eggs, Alevins, fry, and parr, have contributed to the overall declines in productivity in the recent decades.
Okay. And contaminants, as we've already talked about earlier, when we see contaminants on this line, that's referring simply to the measured contaminants that were in technical report 2?
Correct.
So it does not include endocrine disruptors or emerging contaminants?
Correct. One would come to the conclusion that no conclusion was possible.
And pathogens, I think you've already described that there. Why do you say there's no assessment of likelihood is possible for that one?
Could you repeat that?
For pathogens,
Yeah.
why is no conclusion possible for the...
Because there are, it says few data, but there are essentially insufficient data for assessing the exposure of sockeye to disease, as explained in Dr. Kent's report.
And I think you may have already answered this, but I'll just for clarity. Well, these are not, where it says that it's under the column likelihood, it says it's unlikely. That's unlikely to be a primary driver, but it doesn't necessarily exclude impacts of those stressors on other life stages. Is that right?
That's right. They could be contributory factors.
The next life stage is 2, smolt outmigration. And this is one where you indicated you had a lower level of confidence in the data. And why is that?
Basically because we don't have estimates of survival from the time that smolts leave a rearing lake to the time they get to the estuary. So it's hard to know how that life history stage has changed over the period of interest.
Okay, so the conclusion, you did a similar table for that life stage like the other ones, and it's on page, is it page 50? No, before I get to that table, I had a couple of other comments I wanted to make, sorry. Section 4.3.1 talks about plausible mechanisms. I think we've reviewed now what that means. That's on page 50 there. And at the last sentence there, you talk about earlier out-migration could lead to a mismatch between the arrival of salmon smolts in the Fraser estuary and Strait of Georgia. You talk about earlier out-migration leading to a mismatch. Is that also possible for late out-migration as well? Are you excluding later out-migration?
Both could be a problem. Any mismatch could be a problem.
And I think you have an errata on this page as well, page 50. The last paragraph, 4.3.2, exposure of Fraser River sockeye to stressors. So the errata sheet's on the page. Perhaps it's easier if we just have the text of the document. Thanks. You see in the 4th line down, it says they generally spend only 2 months in stage 2 migrating downstream the ocean.
Right, that was incorrect, and that was basically, that 2-month period was meant to describe the period within which all Fraser stocks might be migrating. Actually came from the contaminants report. So it's obviously much less time that each stock spends migrating downstream.
It's more in the range of 7 to 10 days.
Right.
Now sorry, I'll skip around a little bit. We looked at the table 4.3-1, which I don't think we need to spend a lot of time on. That we've identified, I take it the same explanation for other evidence would apply on this table. It's at page 54 of the report. And the same analysis under the likelihood column, although it's for a different life stage. Is that right?
Yes.
Okay. And same for contaminants. It again relates only to the measured contaminants. Is that
correct?
Right? Okay. So we'll move to life stage 3. So this is coastal migration and migration to rearing areas. That begins on page 55. Plausible mechanisms that you set out at the bottom of page 55 and over to 56. We've heard in these hearings evidence of harmful algal blooms, and I don't see that listed under this section. Is that a plausible mechanism as well for declines in the coastal migration and migration to rearing area phase?
Yes, it is. That was an oversight. We do mention it later on in section 4.7 of our report, page 88, but it should have also been mentioned in this section here.
And then page 64 of this section, you look at conditions in the Queen Charlotte Sound and the Strait of Georgia, and you can compare them. You did some additional analysis of data in this section, is that right?
Yes.
Okay, so what was done?
So maybe you could go to page 67. Actually, let's look at page 66, I'm sorry. So we assembled the data that were available for various time periods for the Strait of Georgia and the Queen Charlotte Sound, and the reason for doing that was to explore, again, this was a preliminary analysis of which variables were best correlated with the changes in overall life cycle productivity. And we also included information in these analyses of the spawning abundance of all these stocks as well. So the variables that you see listed here, the last column in table 4.4-1 shows the start of the available data. So we used, for this analysis between 1969 and 2004, we used anything which started before 1969. So that excluded chlorophyll measurements for both Queen Charlotte Sound and Strait of Georgia, and also average sea surface temperatures in Queen Charlotte Sound, which started later. So we included the other variables. And now if you go on to the next page, there you can see the variables that were included in this time period. And the ranking of the model at the bottom is the relative degree of support of that model in the data. That is the relative ability of each model to explain overall patterns of changes in sockeye productivity across all of the, actually it was 18 stocks we included here, we left out the pit. And so the top three models here all had, actually if you could stay on the top three models all had a relatively similar level of support. And the top ranked model here was Strait of Georgia temperature, the second one was Queen Charlotte Sound salinity and discharge, and the third were all of the variables. So basically our conclusion from this is there isn't a clear difference in the explanatory support between conditions in those two regions over this time period.
Were there any shortcomings in the data available for use there? For example, were there things missing in the Queen Charlotte Sound data that you had for the Strait of Georgia data?
Right, we're missing temperature for Queen Charlotte Sound for that analysis, and we're missing chlorophyll for both.
Okay. And then the next table down, 4.4-3, is the time frame 1980 to 2004.
Correct. And now we have the temperature data. We still don't have the chlorophyll data, and so it's basically similar to the previous analysis. In this case, the top three models, you can see the ranking at the bottom there, were all including Queen Charlotte Sound temperature, which is negatively correlated with sockeye productivity, and salinity, which was positively correlated across all of those top three models. And the Strait of Georgia models for this time period showed relatively little support, in that they were lower ranked models, significantly lower.
Okay, and then if you turn the page and you look at 4.4-4, and one page over again is 4.4-5. This is where you compare the importance of chlorophyll in the Strait of Georgia and the Queen Charlotte Sound, the importance of that measurement in explaining the productivity of Fraser River sockeye from 96 to 2004. Is that right?
Yes, that's correct. Now that's a shorter time period, and because it's a shorter data set, we weren't able to compare the relative importance of Queen Charlotte Sound variables and Strait of Georgia variables in one analysis. Otherwise, we'd have too many parameters for the length of the data set. So we just looked at them separately. And so within the Queen Charlotte Sound, chlorophyll was very important. It was in the top 4 ranked models, and so that was interesting. And then within the Strait of Georgia, salinity was the most important in the top ranked Strait of Georgia models, and that was negatively correlated. So the more saline it gets, the worse it was within this time period. Again, it may be different within a longer time period.
And then are you familiar with the work Jim Irvine has done in relation to chlorophyll A levels in Queen Charlotte Sound and Chilco productivity?
Yes.
Right. And how do the conclusions that Jim Irvine has drawn relate to the work that you're showing us here on Queen Charlotte Sound and the importance of chlorophyll?
So they're generally consistent in the overall conclusions, and they're somewhat different in the details. So I'll just quickly run through some of the differences. They don't really affect the consistency, but they're important. So Jim used Queen Charlotte Sound chlorophyll for the first 3 weeks of April and related it to the marine survival of Chilco sockeye only. We used chlorophyll data for April and May, plus other variables such as discharge, salinity, temperature, and spawners to attempt to predict the overall productivity for all of the Fraser stocks, not just the Chilco. So it's interesting that despite the fact the analysis was structured quite differently, we came up with similar results. In the 6 models that used chlorophyll, the productivity was positively related to April chlorophyll in all 6, and it was positively related to May chlorophyll in 5 out of the 6 models. So it's pretty similar outcome to Jim, a different type of analysis.
In this inquiry, we had some reports prepared by Dr. Beamish and others of his group, and they were entered into evidence in July, in our hearings in July. Have you read those reports?
Yes.
All right, and do those reports change any of your conclusions?
No, the main conclusion that's relevant here is that marine conditions have a significant effect. Marine conditions during the coastal migration stage have a significant effect on declines in Fraser sockeye productivity, and that's consistent with what the reports by Dr. Beamish and his colleagues found. And whether that mortality occurs in the Strait of Georgia, which Dr. Beamish has mostly focused on, or in Queen Charlotte Sound, which Dr. McKinnell focused more on, doesn't affect our conclusion.
Right, and I'd just like to go to the table for this life history stage, which we've, similar to the ones we've already looked at, page 70. Now just to, this is referred to in the errata sheet, but please don't put the errata sheet up on the screen. We'll just ask Mr. Marmrick to explain it. You see under the correlation and consistency column and under the other evidence column, there's a little dash, which we haven't seen before, and no data. What do those mean?
So if you recall the decision tree that I described earlier, figure 3.3-3, the dash means that we didn't get all the way through that decision tree to that column, and therefore it didn't need to be analyzed. So in the first row of table 4.4-6 for pathogens, there were not enough data on exposure to pathogens to merit going to examine. You can't correlate data you don't have.
Okay. All right, and then for predators, and to contrast with pathogens, how does the no data column then compare to the little dash under the pathogens column? What's the difference?
So we do have some data on predators in terms of, for some predators, in terms of how they've changed over time, both in freshwater and in the ocean, and have included those data. However, there were not any, until we did our own statistical analyses, there weren't any correlational analyses in technical report 4, was it, I believe, on predators? I can't remember. No, which
9.
is 8. Oh, okay. 8.
Right.
Technical report 8. Anyways, Billy Christensen and Andrew Treit's report. That's easier for me to remember than the number.
The likelihood column, as we've already talked about what that means, and I just note at the bottom of the very last line before the heading 4.47, it says that the conclusion is thus that it is very likely that marine conditions during coastal migration life stage of this coastal migration life stage contributed to the poor returns observed in 2009, and the very likely is contrasted with the likelihood column that we see there for marine conditions, where it just says likely. What's the distinction you're drawing there?
So in the table, we're talking about the overall declines in productivity over 20 years. And in that sentence that you just quoted, we're talking about what happened to the returns in 2009, for which there's a lot more evidence.
And then we see both marine conditions and climate change on that table, and we've heard a lot about both of those. Are they mutually exclusive, or do they actually overlap in some ways?
Well, they overlap in a lot of ways, and that's discussed in technical report 9, I believe, by Scott Hinch and Eduardo Martins. So they talk about how climate change can affect conditions in the ocean in terms of food availability. Also, technical report 4 talks about past changes in marine conditions and temperature, and looks at future changes in marine temperatures with climate change, and discusses how some of the extreme past temperature years look a lot like the expected future years, say in 2080. So what we have is an overlap there where climate change is likely to increase temperatures, and increased temperatures are likely to be bad for food production and changing the kinds of predators that sockeye are used to, all of which is not good for Fraser River sockeye. Now for Alaska sockeye, a little increase in temperature can be a good thing.
The next life stage is 4, which you describe as growth in the North Pacific and return to Fraser. So where does this life stage begin, just physically? Is it at the end of Queen Charlotte Sound?
I think, once the fish get up into the Gulf of Alaska.
Okay, so up past Hackett Strait.
Mm-hmm.
All right, now this one would appear to cover 2 different areas. There's the coastal area in Alaska, but then also the North Pacific, and it's the return home. So is that right, first of all?
That's correct.
Okay.
Until you get to the mouth.
Okay. So is there a difference in the amount of information that's available to allow an assessment, on the return to the Fraser, that sort of return journey versus the growth in the North Pacific phase of their life? There's sort of 2 different parts to it.
Yes, we tend to have more information once they get close to shore. So for example, in the report on page 75, we have the percent of salmon that are returning by different routes, like via the northern diversion or the Strait of Juan de Fuca. And so we get more information primarily from test fisheries as the salmon get close to home.
And this is a life stage where you feel you don't have a lot of confidence. There's not a lot of data available for you to assess. Is that right?
Yeah, it's a big blue box.
Okay. Your conclusion box is set out on page 79. I think we've clarified now what all of the different marks mean in this box, so I won't take the time to do that. But with respect to the different factors set out there, there's nothing for contaminants. Is that because the report by Don McDonald didn't include marine contaminants?
That's correct.
So can we assume by looking at this report that marine contaminants are a non-issue?
No, you can't assume that. It would fall into the same part of the decision tree as pathogens or unmeasured contaminants like endocrine disruptors that you mentioned earlier.
The last stage is the migration back to spawn, and this stage is the stage you feel you have the most confidence in the data for. Is that right?
Correct.
Okay, and that begins at page 80. Page 85, I think there's just an errata correction you want to make. Under the conclusion section, the first line, I think there's a correction you want to make there.
Right. You need to replace life history stage 2 with where it says smolt migration from rearing habitat to Fraser estuary with life history stage 5 migration back to spawn. I was guilty of copying and pasting.
I think we've all fallen victim to that. All right, now in this conclusion table, you have a different conclusion for pathogens than you have previously. In the previous life histories, you've been noted that there was not evidence to support many of these analyses. But here on the return through the Fraser, return to the spawning grounds, you now say that there is some data. Well, what are you referring to there?
So under the other evidence column, as I was talking about earlier, there have been very extensive studies by Scott Hinch and Dr. Farrell and many others on the health of fish as they're making their way home to spawn, and levels of disease, physiological condition. So, and those are field studies, and they're quite well correlated with survival measurements from tagged fish, radio tagged fish. So there are a lot more data for that life history stage.
And then under likelihood column for climate change, temperatures and en route mortality, you have 2 conclusions. One says definitely and one says unlikely, which appear to be radically opposite. Can you explain what that means?
Yeah, so these are very important footnotes. So if you take the definitely first, it has footnotes B and C there, which means harvest and escapement. So the en route mortality, and there's graphs in here that were drawn from the report by Martins and Hinch, shows that en route mortality has increased over time and has been very substantial, particularly for the early run and late run sockeye. And so that affects the number of fish that get back to the spawning ground, or would if you have the same level of harvest, and harvest has been reduced, accounting for the expected en route mortality. So there's definitely an effect of climate change and temperatures on both harvest and escapement. However, as I mentioned earlier, the way in which recruitment is measured is escapement plus harvest plus en route mortality. So it's essentially the number of fish that return to the Fraser prior to any harvest or prior to any en route mortality. So that declining productivity cannot be explained by en route mortality, because en route mortality is already included in it, already included in recruitment. In fact, as en route mortality goes up, recruitment would go up. So it's unlikely to be an explanation of declining life cycle productivity.
And then the addendum, which has now been marked, or was earlier marked, I should say, in these proceedings as Exhibit 1575. You have a similar conclusion table in that document, and if that's at page 18 of the report, kind of spills over 2 pages. There, okay. And now again, you have 2 contradictory notations for disease of salmon farm origin, and you'll see that on page 19 there. It says possible and unlikely. And can you explain what you're talking about there and how those can they be reconciled?
So our job here was to look over the work that Dill and Noakes did and look at what the implications of their conclusions were for our overall conclusions in Technical Report 6. It wasn't our job to try to reconcile them or read the 250 references that they referred to, only 25 of which they looked at in common, by the way. Anyway, and so we basically said, well, if you took Dill's report as evidence, your conclusion would be that disease from salmon farm origin was a possible contributor to the overall declines in sockeye salmon, and if you took Noakes' report, your conclusion would be that that was unlikely.
Okay. And then just one last figure before the break. The figure 3, in which if you just scroll down, Mr. Lund follows this table. What is that indicating to us?
So as it says at the bottom, these are the mechanisms identified as possible, or no conclusion possible, based on Dill 2011. So grayed out in this figure are the hypothesized mechanisms that both authors cumulatively were looking at in terms of causal pathways by which sea lice, waste, escapees, and disease could potentially affect sockeye salmon from salmon farms. And so the ones that are, that was the overall diagram, and then the ones that are grayed out are the ones which were considered to be unlikely, and the ones that remain are those that Dill considered to be possible, whereas Noakes considered all of those pathways to be unlikely.
Thank you. Mr. Commissioner, we could take the break now if that's convenient.
Hearing will now recess for 15 minutes.